So my name is Suzanne Jin, and from the CRG at Barcelona. And today I'm going to give this talk that I prepared together with Susanne Jodoin from Quebec. Now, the talk itself is about the differential abundance pipeline, but it's also about the following question. So, given the same task, there are often multiple alternative methods, and each of them may provide different answers. Then, what do you trust? And how should we account for this when building a pipeline? Now, this is the main thread that I want to talk in this presentation, but before that, let me first introduce you about what is differential analysis through a case study performed by Quebec. Now, many of you may know somebody that has diabetes and they have to inject insulin every day. But what if there is an alternative treatment that can avoid that by transplanting healthy cells so that the body can auto-regulate the levels of insulin? Well, the therapy is very promising, but the bottleneck so far is that many transplanted cells die due to the low oxygen conditions after the transplantation. And because of this, this case study, what they want is basically to find out if specific treatments can actually help the cells to better survive, and if so, how it's done and what are the mechanisms underlying that. For that, they compared the treated conditions versus untreated conditions, ask what are the differences between them. Here is where differential analysis can help. And basically, if given gene expression data, what differential analysis tries to find out is which are the genes that have expression levels that are changing across the different conditions in significant ways. This pipeline includes some of the existing tools for this kind of analysis, and therefore, when Quebec used the early version of this pipeline in order to find out the kind of question they are asking, then they find out that actually specific genes and pathways related to glucose metabolism are actually upregulated because of the treatment. And that might be the reason of why this treatment is helping pancreatic cells to survive and maintain its functionality. Now, obviously, this is just one case study, but in general, you can ask the question of what are the differences between any conditions, such as healthy versus disease, tissue one versus another tissue. And it can also provide, it can be applied for different purposes, not including generating knowledge, but not only that, such as also for perhaps finding out candidate genes that can be used for biomarker discovery or for therapeutics and so on. So a lot of important applications rely on differential analysis, and therefore it's extremely valuable to get the answer right. However, in practice, this is not always that easy in many cases. To understand that, let us focus on the top used tools, which are Dissect2 and ETSR. They together are cited more than 100,000 times, and this means that they have been very useful in many cases, and they are quite popular. Yet, this does not mean that they are going to be useful for all the kind of cases that you can imagine for. Indeed, what this study, and maybe perhaps more studies, find out is that both methods are producing high false discovery rate when the dataset has a lot of samples. And as you can see from this figure, that shows the number of genes that are identified as differentially expressed by different methods from randomly permuted data. This means that expectation is that none of the genes should be actually significantly different across the conditions because everything's random. But despite that, both methods are finding out some 100 genes to be differentially expressed. This means that they are basically just discovering false positives. Therefore, they are not suited for this specific case, and maybe other methods will be better fit. Now, up to here, you may ask, what are the specific reasons that are driving this? Well, in part, this is because different datasets have different properties in terms of noise level, batch effects, sparsity, and even the size of the data. And so far, there is no universal method that can be used to cover all these different aspects together and provide a robust answer for all the different cases. Instead, each method has been developed are addressing specific needs, targeting specific data types, considering specific assumptions about the data, and using specific combinations of methods for normalization and statistical testing. So there is no one-size-fits-all solution. Then how can we actually account for this when creating a pipeline? To address this, we decided to convert this pipeline into a hub pipeline. Here I want to highlight that we are not the only ones creating hub pipelines. Instead, any bioinformatics task can be potentially, tasks that can have many different alternative methods, can potentially be used to create some kind of hub pipeline. This is the case for differential abundance. This was also the case for the multiple sequence alignment pipeline that was created within our lab at CRG by Luisa Santus, and it will be the case for many other more pipelines such as protein folding. Now, after here, you may ask what is exactly a hub pipeline, what makes it different from a standard pipeline. Well, basically, what we want to offer with hub pipeline is first that the users will be able to accelerate discovery in a way that is not constrained by a specific method, because they will be able to choose between different methods. And not only that, given all the different methods be in one centralized place, you can imagine many additional features that can be enabled by this, right? These can, for example, be the possibility to run and compare different methods and then choose the proper ones for the use case, or even combine different methods for some kind of consensus and then use that if the performance is better. And if you were a developer, then you can also run all the different methods in just one click and then use that to benchmark against your own method and therefore accelerate development. And eventually, if development is in coordination with such a production framework, then the contribution of such a method afterwards will be also more immediate, and that will also help method accessibility for the users. So in a way or another, this is more kind of like a ecosystem or some kind of like that you can imagine through. And it's quite ambitious to achieve all these features all together. A lot of work has been done, a lot are still in progress, but thankfully there are actually many collaborative efforts that are in one way or another going all towards this direction. So the pipeline was originally created by Cubic because of their high need for doing this kind of analysis and high throughput of this data set as a core facility. But along the way, they have been a lot of different developments by different people who have been shaping the development. This includes Jonathan Manning, and also the people from BI who have been progressively adding also alternative tools to the pipeline. And here we are from CRG, that we eventually wanted to introduce this idea about the hub pipeline. So a lot of effort were put together, and all this shaped this pipeline to get closer and closer to this vision of hub pipeline. I will not get into too much about specific details, implementations, but let me first introduce you a brief overview about the pipeline and highlight key features. So the pipeline looks something like this. It has 4 stages, basically. It starts with some data validation, featuring, preprocessing. Then it runs differential analysis. The outcome of this step is then used by functional analysis to provide some sense of what are the functionalities and pathways that are actually affected. And the end step will provide a report to gather all this information together. From here, I want to highlight 3 key features. First, the pipeline do not only provide alternative tools for the key analysis, but it also can take in different kinds of omics datasets, such as RNA-seq, microarray, and proteomics. Then, all the methods that belong to the same task or address the same task are actually encapsulated into respective hubs workflows. This is thought in order to so that the switch between different tools for a given task can come more naturally, and then in the future the addition of alternative tools will require minimal disruption to the main pipeline code. And beyond this, I want to highlight one last feature, which is in order to enable the pipeline to be a hub, a true hub that can allow users to explore different tools and different analytical options, we provided a way for the pipeline to run different paths across the entire pipeline in parallel. This means that as a user, you can run your favorite tool, many different analytical alternative tools, or even different analytical paths that use different combinations of tools and pipeline parameters. And this is what actually took us quite a bit of effort, because it's quite different from what the other pipelines usually do. So in a standard pipeline, a channel usually defines the flow and the stream of data through the pipeline. Here, what we changed is that we extended this flow to not only include data, but also pipeline parameters. And in this way, the pipeline can just take one set of pipeline parameters, another set, and more, and parse all this into respective channels and run all the different options of analysis in parallel. Now, to finish the talk, I want to say these are not this is not the only thing that is needed for a true hub pipeline. Instead, there are still a lot of ongoing effort to make the pipeline going towards this direction. And this includes, for example, extending the scope of the pipeline with additional tools. And this is actually getting done by many people who are interested in alternative methods. Then in parallel, there's also this effort by Júlia, who is creating this NF class that can eventually help to extend the pipeline in more automatic ways, because the idea is that you will be able to automatically classify and incorporate the models into the respective hubs, workflows, and pipelines with the help of agentic AIs and CI procedures. Now, obviously, there's still the implementation that we have is still quite immature, so there's a lot of ongoing work in improving it, and there are many mechanisms that are required for consensus, and comparing methods are still on the agenda. So a lot of ongoing work, and therefore if you are interested in any of these, please join us and also get into contact in the hackathon. And thanks a lot to everybody that contributed to this pipeline, especially to those from our lab at the CRG, but also to people from Cubic, especially Suzanne Jodoin for preparing this talk with me together, and also Jonathan for all the help that he provided in order to make this pipeline actually closer to the hub vision that we have. So also thanks to you for listening, and yeah, that's it.